Welcome to another discussion of fermentation. Today's subject, beer. This is a slide that I've opened with in previous lectures, and so it's worth pointing out how much we have to think about fermentation when we think about different kinds of foods. We've discussed yogurt, we've discussed kimchi, we've discussed soy sauce, we've discussed sauerkraut, we've discussed acetic acid. Previously, we've discussed wine and breads, right? Yeast leavened bread. So we see how important these bugs, these germs, these microscopic organisms are to good food and to so many different kinds of food. That's the reason that I focus so much on fermentation. And I think you'll agree that our food culture today is very focused on cleanliness and the exclusion of microorganisms from our food. But in fact, we owe a lot to those organisms, and a lot of the good food that we enjoy today, we would not be able to in the absence of those bugs. Okay, preaching over. The fermentation, this is derived from the Latin word that means to boil. In this case, we're not talking about water vaporizing because of heat. We're talking about CO2 forming, right? Carbon dioxide forming as the yeast ferments the sugars, and those bubbles form and bubble away. So that's the seething. That's the internal commotion that takes place during fermentation. What we're going to focus on today is the leaven, and that is yeast. Saccharomyces cerevisiae, right? Acting on the saccharine liquid. Saccharine, what does that mean? We're just referring to sugars. The liquid that contains sugars. I want to point out again to you this name of the scientific name of yeast, Latin name of yeast, Saccharomyces, related, of course, to the substance upon which the yeast acts, the sugary liquids. Won't belabor it too much more, just to remind you that there are many different kinds of fermentation that take place. While we're going to focus on alcoholic, beer, wine, and the distilled products thereof. Stay tuned for that. We've also talked about vinegar already. We have talked about lactic fermentation in a couple of different contexts, but let's just write down yogurt. So we've seen fermentation in many different places. All right. What is fermentation? Okay. Let me just preface this slide and many other slides to come where you're going to see these sort of organic chemical structures of molecules. I don't want you to panic if you feel like that's beyond your skill level. I want you to enjoy the ride and realize that there's several layers of complexity here that we can delve into as needed. But bottom line is, fermentation is the conversion of these complex sugars ultimately to ethanol in terms of beer and wine. What is a complex carbohydrate? Well, here I've drawn something that actually isn't terribly complex. This is lactose. It's a dimer. That means it's made out of two simple sugars latched together through this glycosidic bond, through this oxygen here. It's a dimer. The first monomer here is galactose. Galactose. The second monomer is glucose. Those of you that care will remember that galactose is linked to glucose and lactose through something called a beta 1 to 4 glycosidic linkage. Here's carbon 1 of the galactose. Here's carbon 1, 2, 3, 4 of the glucose, and they're linked through that beta linkage. We won't go into what beta means, but those of you that are interested in the biochemistry, go find out. Anyway, a complex carbohydrate. Now, really, when we talk about fermenting, whether we're talking about bread dough or whether we're talking about malted grains when we make beer, we really start with something much more complicated, which is starch. In many cases, starch. Now, it's simpler than lactose here. Starch is simpler than lactose because it's mostly made out of glucose. It's entirely made out of glucose, in fact, in these very complicated and large polymers. So we have to degrade that starch into something that can enter a biological pathway, a metabolic pathway known as glycolysis. Glucose enters glycolysis directly, and the end point of glycolysis is pyruvic acid. I've written this as pyruvic acid. Again, those biochemists in the audience are going to say, well, don't we usually say pyruvate? Yeah. So here's the pyruvate structure. Right? Conjugate, oh, that's cute. Conjugate base of pyruvic acid. That's our end point of glycolysis. Now, it's not a very good end point for the yeast. We can still get more energy. Of course, that's what glycolysis is all about, is getting energy out of our sugars. There's still more energy to be got out of pyruvic acid. Let's come back to that in a minute, though, and just point out that sugars like lactose are made of other stuff besides just glucose. There's also galactose, and for sugars like galactose, there's a whole other slew of pathways that we won't talk about that can lead us to glucose, or that can otherwise allow us to enter glycolysis to get down to this fundamental molecule of pyruvic acid or pyruvate. Okay, great. What happens next? That's what's really interesting today, reminding us that our goal is to get to ethanol, right? And so here we see that we can break down pyruvic acid ultimately to ethanol through acetaldehyde. And let's look here at what the organism that's taking that job on for us. That's going to be our yeast. Here I've given you a key. There's that Latin name again for brewer's yeast, Saccharomyces cerevisiae, saccharin. Okay, it's a fungus, and a simple organism, but not so simple as this other fermenter, which we've encountered before as well, lactobacillus. Lactobacillus, of course, taking pyruvic acid down a different pathway to the production of lactic acid. So, a few different pathways that we can take. All of these, again, aimed at deriving a little energy, a little more energy out of the sugars that we use to ferment. So what else can we say here? Fermentation takes place under anaerobic conditions. Remember, that's no oxygen. Well, okay, fermentation takes place under conditions in which we lack oxygen. Now, that's not always true. Yeast is not what's called an obligate anaerobe. It can also ferment to ethanol in the presence of oxygen, but that's another story. Of course, in working muscle, there's another process that takes place, and we're the anaerobe, we're the fermenter in that case, if you want to think of it that way. We won't belabor this discussion today, just to remind you that we convert pyruvate to lactic acid in our own bodies to allow glycolysis to proceed. So I'll just draw a little dashed line here. The point of the lactic acid, again, we're not belaboring that, is to allow glycolysis to proceed in the absence of oxygen. Minus oxygen. So that's fermentation. One other detail I can remind you of, which we talked about previously, there's another bacterium called Enococcus. Sorry about this. Enococcus, right? That converts malic acid to lactic acid. That's an important step in the production of certain wines, the malic fermentation. But that's something we've discussed already. We won't return to that. But again, big picture, conversion of sugars to something wonderful, ethanol. Okay, let's talk a little bit about what ethanol does in the body. So, this is the reverse of what's taking place in the fermenting bacteria and yeast. We've got ethanol. We're starting with ethanol. This is a toxin. Toxic molecule, has lots of depressant effects in the body. We won't talk a lot about those right now. Instead, let's just keep it chemical and say that the ethanol is degraded by two different enzymes. These are enzymes. Alcohol dehydrogenase first takes ethanol, pops off a CO2, right? Whoa, and converts the ethanol to acetaldehyde, and we get a little reduction of NAD plus to NADH. For those of you that don't care about the biochemistry, there's niacin, one of your B vitamins. But the second step is also interesting. We have to take that acetaldehyde, which is otherwise also kind of toxic, right? Or has some ill effects in the body at least. and converts it to the innocuous acetate. This enzyme is called acetaldehyde dehydrogenase. Again, those of you that are interested in little biochemistry, these dehydrogenases tell us something about the other molecules involved in the process. Anytime you see an enzyme called a dehydrogenase, you can be pretty sure that NADH or something like it is involved in the chemical process, in the biochemical process. What else to say here? So these are enzymes. They exist as, both of these exist as isoenzymes that vary from population to population. What does that mean? Well, it means that different, let's say, we'll use the terrible term race because it doesn't tell us a lot about the genetics, especially in certain cases. But we'll say racial groups can express different kinds of alcohol dehydrogenase, different isoenzymes of alcohol dehydrogenase. And what this can lead to is differential abilities for processing that alcohol into that innocuous acetate, right? So that some isoenzymes will prolong the effects. I'll write that down. Prolong the effects of the ethanol because of their inability to break it down. Another really interesting set of isoenzymes are in the acetaldehyde dehydrogenase family, are responsible for something referred to as Asian flush. You may have seen this if you yourself are of Asian descent or you know someone of Asian descent. Relatively small amount of ethanol can produce a very noticeable reaction on the faces of Asians, and that is bright red spots appear on your cheeks. The cheeks appear flushed. That's because they are expressing an acetaldehyde dehydrogenase isoenzyme that's ineffective at converting acetaldehyde to acetate. So you have a buildup of acetaldehyde concentration. That's what this little symbol means, right? The non-functional acetaldehyde dehydrogenase and isoenzyme results in an increase in the concentration of acetaldehyde, has these sort of negative effects. Let me just point out, too, that the study of these isoenzymes is a hot topic. People want to understand, scientists want to understand, society wants to understand why some people respond differently to alcohol consumption than others. And while there are a lot of factors involved in individuals' responses, a lot of it is bound up in what kind of isoenzymes of these two different enzymes you're expressing. What else can we say about intoxication? Okay, so here's a nice figure that I stole from this paper, Journal of Pharmacokinetics and Biopharmaceuticals. It's an old paper, but it shows something very nice, which is the blood alcohol concentration expressed as milligram percents. I'll come back to that in a minute. Basically, it just means milligrams. I'll abbreviate ethanol for this. Milligrams ethanol per 100 grams or milliliters of blood. Okay, so what we see here as a function of different numbers of drinks consumed, we look at the amount of ethanol in the blood as a function of time. And what we see for one drink is that we come up to this pretty small level, and then after a couple of hours, the ethanol leaves our system. Again, exiting the system as acetate and carbon dioxide. And we see that alcohol level pop up if we drink a second drink, again as a third, again as a fourth. And we see that with 4 drinks, we really take significantly longer amount of time to get back down to 0, right? 2 hours for 1 drink, 7 hours for 4 drinks. And these are relatively small drinks. Why am I pointing that out to you? Just to give you a sense of what happens to the blood alcohol concentration as you consume more and more alcohol, and then as that alcohol exits your system. Let's just make it clear, however, that this blood alcohol concentration metric isn't the one that you're used to seeing. What you're used to seeing is the blood alcohol expressed as a regular percentage of alcohol by volume of the blood. And we're used to seeing these numbers, 0.1 to 0.3. That's a percentage, 0.1% alcohol. That's referring to somebody that's pretty intoxicated. And in fact, the legal limit for alcohol percentage in the blood is 0.08 for someone 21 years or older in California.